So Bismillah, salat salam ala Rasulillah. Today, inshallah, we will continue or complete the 3 remaining slides from yesterday. We've talked about the types of hypersensitivity, and we mentioned it's the mechanism underlying one of the diseases of immune system, which is the allergies. Okay. So we've reached type 4, and we've discussed, if you can go back, we've discussed the mechanism, and we mentioned that it's either mediated by direct killing by cytotoxic T cells, and this is in the example of tissue rejection when you are transplanted with a foreign organ. The CTL will carry direct killing, which is called graft rejection. So your immune system or your body reject the newly transplanted organ, or it can be mediated indirectly through the helper CD4 cells, which somehow activate macrophages with the secretion of certain cytokines. Then these macrophages will amplify the immune response by release of a variety of cytokines or inflammatory cytokines. Okay. So to continue, it's kind of, we mentioned that the other type is somehow close to type 1. What do they attack, Hussam? They attack our, the newly, if you are transplanted, يعني نقلنا لك عضو من شخص آخر، فما حيكون يعني completely compatible. حيكون في شوية differences. Certain foreign antigen will be presented. But CTL directly attack those organs. Okay. We mentioned the 2 indirect way mediated by T helper. Yes, will not be acceptable. So your body try to reject that. Delayed type hypersensitivity or contact, which is the one that you it was explained in the type 1, which is mediated by IgE. But for type 4, mostly we are talking about this, and the antigen here is in the form of proteins. So for example, you've been injected with a venom, يعني one of the venom here, insect venom, اللي هي الtoxins, أو mycobacterial, one of the bacterial species protein. This one can stimulate the initiation of type 4 hypersensitivity. Okay. The consequences, local skin swelling, as we will see inshallah later مع البكتيريا, arrhythmia, induration, cellular invertebrate, so there will be some sort of skin inflammation. All right. This one mostly associated with type 1, we said, but also somehow overlap with type 4 hypersensitivity. And the antigen here is not protein, it's not a venom, but it's hapten. So a chemical, all right? Poison ivy, and this is one of the plants. Mango sap, العصارة اللي بتطلع من المانجو. Dinitrofluorobenzene, it's also a chemical. So these chemicals, of course, another like nickel and chromate. Nickel, بعض الناس لما يلبسوا, when they wear watch, يعني الساعة بتكون فيها nickel with contact with your skin. Some of you guys somehow irritate their skin. Their skin will start to itch. It will start to become red and hot. And this is an indication that there is type 4 hypersensitivity taking place, because the nickel will infiltrate the skin, will invade the skin to go underneath and stimulate an immune response. And of course, again, somehow similar consequences, epidermal or dermal reactions. Okay. More details onto the mechanism of type 4, and this is an example of tuberculin skin test. Not sure if you heard about this test. هذا اختبار اللي هو you are tested whether you have got Mycobacterium tuberculosis before or not. اوكي. وهذا بيسووه يعني in the hospital when you start يعني your practice in the hospital usually they do these tests, particularly يعني لو you are practicing in infectious disease unit, you need to be tested on this. Why does it considered type 2? Which one is type 2? No, this one is type 4. We are talking about type 4. Hapten, since there is a hapten, well, that's the things, because we said there is an overlap. يعني في تشابه شوية, تداخل ما بين type 1 و type 4 in this particular type of, but لكن mostly قلنا إنه لا, يعني خلينا نرجع mostly هذا. okay. Some of the times maybe this also اللي هو بيشبه type 1, لكن let's focus on type 4, this one. okay. So more into the mechanism I said. So antigen is injected to subcutaneous tissue and processed by local antigen presenting cell. We have cells. The antigen presenting cells is all over our tissue. There is in the brain, in the liver, in the lung, and in each place they have different name. In the skin, if you remember, it's called Langerhans cells. Antigen presenting cells in the skin is called Langerhans. In the brain, microglia. In the liver interstitial. In the lung interstitial. In the liver stellate cells. So we have a lot of, yes, they are all dendritic cells, but when they go to certain tissue, they have different name then. فإن the skin we call it Langerhans cells. اسمها Langerhans, the guy who discovered it. So these are the antigen presenting cells. They will يعني receive or bind to the antigen that was injected through the skin. Of course, then it will stimulate immune response through T helper cell. See, T helper from previous immunization. You have memory. Now what will happen? Th1 effector cell recognize this antigen. I saw this before, and release cytokines which act on vascular endothelium. Why? Because we need to bring more cells to fight, as it's happened here. Now, the permeability of the endothelium will be يعني separated, and then we will have the cells infiltrate. It leaks from the capillaries and bloodstream into the skin tissue, and then we will have this form of induration like this one. It's shown in the skin. So we have a lot of cells, a lot of fluids, a lot of cytokines come and accumulate, and then we will have this blister filled with fluid, and then we measure. طبعا في التيوبر كلين test we measure the diameter. Depends on your scale. At certain threshold you are positive or you are negative. You will get the details in the next years, inshallah, about this particular test. So that's what happened. And here kind of representation. This is antigen presenting cells. It talks to the T helper. It tells her, hey, come on over here. I have antigen found. And then T helper will come, and of course it will stimulate other cells to come. More cells will infiltrate the tissue. What is the blue thing? The blue thing here, you mean, or where? This one. These are the T helper. T helper 1. Okay? اللي هي هنا. This arm. T helper 1 stimulate macrophages. Macrophages secrete cytokines, increase the permeability of the capillaries, allowing more cells to come. And the circle is endo. This is, yes, this is the capillaries. So these are the endothelium. See now the permeability increase, allowing the cells and fluid to leak. When they leak, then you see this. All right? Okay, finally, this is the other type. اللي هو بيشبه type 1. اللي depends on contact. بنسميه contact sensitizing agent. Why? Because it happened by contact. Like زي لما تلبس الساعة مثلا. او يجي يدك تيجي على كيميكالز زي المانجو ساب مثلا، ولا البويزن ايفي، هذه بلانت توكسينز. Right away, just with the contact with the skin, right away they get inside and stimulate immune response. All right, so it happened at the same mechanism. Still, contact sensitizing agent penetrate the skin and bind to self proteins, which are then taken up by Langerhans. هذه السيل اللي هي الانتيجين بريزنتنج سيل residing في السكين tissue. تمام؟ هذه antigen presenting cells. These are the toxins or the sensitizing chemicals, okay, the red ones, attached to self protein, and then it's taken up by macrophages. It will, or the ABC, of course. Then T helper 1 will come, activate, produce a lot of cytokines. It will even activate the keratinocytes, which is the superficial layer of the skin, and those will release a lot of sets of chemokine and cytokines. You don't have to worry about these, but just collectively there is an inflammatory response. Eventually the same thing will happen. We'll have a recruitment or leakage of cells and fluid, okay? Activate macrophages to secrete mediators of inflammation. Where does the interferon go? Interferons goes to the to stimulate the recruitment of other cells. Okay. All the cytokines that are released, whether from T helper, whether from the macrophages, they are all initiate inflammation. طبعا هنا, in addition to the leakage of the capillaries and recruitment of macrophages and fluids for the induration to happen, they also stimulate the keratinocyte. Keratinocyte, it's also considered part of the skin immune system. They participate, they secrete cytokines, and that will even aggravate the itching in the skin, because this is the outer layer. So the bottom line, inflammation. How? T helper, minal effectors, T helper, antigen presenting cells, and macrophages. What happened? Leakage, increased permeability, leakage, coming of the cells, accumulation of the fluids, and we have indurations. All right, induration اللي هو الارتفاع ده اللي بيصير في السكن لأنها filled with cells and liquid. So we just need to have the main idea. Exactly. I only focus on the main ideas, on the principle, on the concept, on the general idea. I don't go for details, because I know details, you memorize it today, you'll forget it tomorrow. Right after the exam, they will be gone. But I wanted to understand concept. Now, this is not guaranteed, because it's not the only one I'm putting questions. So there are a lot of faculties in all over the campuses. So you need to be prepared, I'm sorry to say, for some details. But I don't expect someone will ask you about this in particular. I don't expect at all. طيب. Okay, so we are done with this session, hypersensitivity and allergy. Again, try to focus more on the mechanism. What happened? Like these figures are quite helpful. يعني هنا I will focus. We have the effector, we have the location, we have the mechanism, effector mechanism, what happened. And then, of course, you see the representation. We have Th1 macrophage, we have CTL. And again, for all the other types, if you try to focus on these type of figures, I think it should give you all the details that you need. All right? So we will conclude this one. So I will stop the